Well, alrighty, guys, it is time to discuss the great Agar Lab of 2024. Let's start with third period bio. You'll see a lot of these have a complete diversity of different things growing. Not so much these guys, but some of the other ones are just packed full. And again, the word I use is diversity, which means there's a bunch of different species of bacteria, and in some cases mold, which is a fungus. Now, let's zoom in on these guys, okay? You see all those little white puffy things that look like pimples, and some are yellow, some are orange. Those are all different species of bacteria. Whereas on the side over there, that big white fuzzy or hairy looking thing, that's the mold or the fungus, okay? So a lot of these look pretty darn diverse, pretty nasty. In fact, what most of the guys told me is that they simply just swabbed the tops of their lockers, which are dusty and dirty and grimy, so they haven't been cleaned for a while. I know there's a couple of groups that told me they swabbed the lunch tables. Kind of the same thing. There's food, there's crumbs, leftovers, things of that nature. So this agar that the stuff is growing in, that's just the food. So that allows these colonies to grow and reproduce and allow us to see them, basically. We move over to fifth period. Again, more of the same thing. You have mold, a lot of bacteria growing. Same thing, the guys swab the same things, everything from doorknobs to again, lunch tables, tops of lockers. It's pretty nasty in terms of bacterial growth. And I'll show you sixth period down here. That, again, it starts with something pretty nasty, pretty cochino. I don't even know what some of this is, but it's probably everywhere from 12 to 15 different species. And in that cool, you see little yellow bacterial colonies. There's an orange one. And then, most of this is in fact different species of mold or the fungus once again. These guys accidentally cracked through the agar, but it's all good. Things still grew. And I always say it's a little bit like fishing in the sense that you never know what you're going to catch. You throw it out there and, things grow and hopefully it looks cool. And that's what the guys kind of enjoy most of all. Let me kind of mosey on over to my seniors. They did, the same thing. But a lot of these, you'll notice there's not as much growth. It's kind of cool, right? Like look at this first one, look at this one over here. I see a lot of these little white colonies. A lot of this stuff is staph, staphylococcus. And as soon as I say staph, I know a lot of students get kind of alarmed, like, oh my gosh, I'm growing staph. Remember guys, staph is a normal commensal. It's found all over our skin. It really doesn't become problematic unless until you have an infected wound, like if you play football and you scraped your knee, that kind of thing. So I asked the guys, where were y'all swabbing? And a lot of the seniors told me they swabbed their armpits. So like something like this, it looks like I have all sorts of growth, but if that's an armpit, that's actually pretty clean. It's just one species that took over because of the medium, the agar. Same thing, it looks like I have all sorts of things growing. Again, that's just only a handful of species. Like this here, that looks big and nasty, but that's just a mold. That's simply just one big mold. And in terms of biodiversity, it's still just one species. So it's like saying an elephant is big, a mouse is small, but it's still just one species, one elephant, one mouse, same concept. Now, as far as some of the things I did for funsies, my mealworms, I swabbed my critters. Again, it looks like it's nasty, but it's not too many species. My turtle, my radiator slider, a little bit worse. The Sudan plated lizard, as far as my pets go, that's the nastiest. The whole plate's filled up. I swabbed the dirt that my plants are growing in, but it's dirt, like that. It's kind of the point, right? And then the nastiest thing I had was this weird combination when I was swabbing my aquarium, my blinds, the inside of the AC. And again, that's one of those that's like probably 10 or 15 species or more, and it's a combination of bacteria, fungus, mold, what you name it, it's there. But it's the air we breathe. It's stuff that's all around us. It just looks nastier than it really is the moment you give it this medium, this agar, to grow. It's kind of like I said in class, if you're in space, you cannot see an individual person, but you can see a city. And that's kind of the same thing. You can't see individual bacterial cells, but when you make a colony, especially with agar, then you can see it with the naked eye. Finally, you see my experiment side of the lab here. I did my shoe, the sole, and then I cleaned it with vinegar. So I have, of course, a before and after. So did the vinegar work? Yeah, it totally worked. You see how I have a lot of things growing. Some are yellow, some are orange, some are, little white clear things, some are cream colored things, some are mold and big and poofy. I got a variety. I've got a tropical rainforest of bacterial and fungal growth here, you know what I'm saying? Now look at the after, it totally worked. In fact, the only thing growing is that mold that looks like a weird gray colored mushroom or something. So look carefully at that. Do you even see it growing here on the before? Which tells me, or at least I suspect, I have some sort of fungal contamination, probably from the paper towels that I used to dry the sole of my shoe. And guess what? We see the exact same thing here with this gecko lid. You see it's covered with mold and some bacterial colonies on the before. The after is relatively clean with the exception of that one big pink fuzzy mold colony growing there, which tells me that it's probably contamination once again from the paper towels. Well, I have passed 6 minutes on this action-packed lesson, so let me kind of back it out. We'll get one big overview. And that's about it, guys. Hope you enjoyed this. Until next time, peace out, God bless, and see you tomorrow.